go ahead and get started. It is an absolute pleasure to be here to have a chance to introduce David Sleboda on this occasion of his thesis presentation and defense. David came to us from Michigan State University, where he received a BS in zoology, and when he arrived at Brown, he had actually had an interesting set of experiences in undergrad. He had done a little research on worms, and he had helped Dick Hill with some textbook writing, and yet he had actually relatively little experience in the area in which he's now an expert, and that is the mechanics of muscle contraction, although the worm stuff turned out to be more relevant than we realized it would be. And David's been just a wonderful student to have around. He's a fantastic lab citizen, department citizen, a positive presence, and he's also been just an exceptionally good scientist. It's been really fun to watch him grow as a scientist and become really an expert. As many of you know, we have a tradition of first-year projects in the department, and first-year projects are meant to be sort of, I call them sort of science with training wheels. They're meant to be these very nicely constrained, confined, defined projects that are sort of canned. They're meant to be sure to work. And I'm a strong believer in these. And so David and I chatted about a first-year project when we first arrived, and we settled on one that, if I had to pick one word to describe, I would say it was horrible, maybe irresponsible on my part. It was completely open-ended. It was completely undefined. It was essentially, we do isolated muscle studies in our lab. We put the muscle in a bath that's meant to mimic the environment that it sees when it's in the organism. So it's got salts, it's got sugars, it's got some oxygen, keeps the muscle happy. And we had made this observation that if you get rid of that solution and you put distilled water in there, I don't know why you would do that, but if you do that, you get more force out of the muscle. And it was kind of weird. We didn't know what was going on. And the project we settled on was sort of, here, David, figure this out. And somehow that worked out, I think, pretty well. So I got away with some irresponsible advising in this case. I think thanks to David's talent. One of the really hard things in this situation is that I have a lot to say, and this is David's time, and I can't eat into it too much. I could tell you how creative his work is, how rigorous it is, how significant, but I think that'll all be apparent in David's presentation. And so given the limited time, I decided naturally that what I would talk about is mountain biking. So David and I have had a chance to mountain bike a few times over the years, and on one particular episode years ago, at a SICB meeting in Austin, David and I played a little hooky in the afternoon, rented a couple mountain bikes, and went mountain biking. And we ended up on a trail that was a few times, I think more than once. We ended up on a trail where you're in that position, you're mountain biking, and you start to get to trails you just can't cover on the bike, and you're carrying the bike, and you're doing more hiking than biking. And on several instances when we were in that situation, I sort of had my head down, and I was just charging through, and David said, you know, I think the bike trail's over here. And I was just charging through it, and I think that, in this sort of soft-spoken way. And the reason I mention that is I've thought of that example, I don't know if I've told David, but dozens of times when we're working on scientific problems, because I would tend to get my head down and charge into a problem, even when we're running into a dead end, and David would sort of quietly say, I think the interesting stuff's over here. So the reason I wanted to use this particular example is David's work will speak for itself, but what you won't see are the many paths that David started down, figured out weren't the right path, either because of limitations of methods, because of a conceptual challenge, and had the kind of intellectual agility to redirect, come back to the right path. And that takes a lot of talent and insight and intuition. It also takes a lot of hard work, because you have to leave a lot of work on kind of the cutting floor. So with that, I will let David tell you about his thesis work, and it's really fun to be here for this occasion.
Great. Okay, well, thanks, Tom, for that super nice intro, and thanks, everybody, for being here. It really means a lot to be able to present this to so many people that are important, so thanks. So I have lots of people to thank and acknowledgements, but I put those at the end, partly so I don't get too emotional or anything before the science part of the talk. So I'm just going to charge right into that, and at the end there will be time for acknowledgements and hopefully some questions as well. So as you can see, the title of my talk and of my dissertation overall is the hydrostatic skeleton of muscle. And my plan for this talk is I want to start with a really broad introduction to muscle physiology just as an academic field of study to kind of give you some context for what this research is that I did during my time here. And then I want to take a couple minutes and kind of tackle this central term that the whole dissertation is built around of hydrostatic skeleton. So what is that? What are some examples of a hydrostatic skeleton? And how is it relevant to muscle at all, which is really the subject of my work. And then I'm going to get into the meat of the talk and show you some results and conclusions from really two main veins of research that we've carried out over the last 6 years. And all the research that I've done really focuses on one central question, which is, does muscle have an intrinsic hydrostatic skeleton that influences its mechanical behavior? And hopefully what that question means will become more clear as we progress through the introduction. So really broadly to start off, what are muscles from the perspective of a muscle physiologist, somebody who studies how muscles work? So muscles are organs in the bodies of animals. Just like any other organ, they're made up of cells and connective tissues, but where other organs are specialized for food storage and digestion or gas exchange, muscles are specialized for the production of force and movement. So the muscles that we're probably most aware of and familiar with are skeletal muscles that attach onto a skeleton of some sort, and they allow us and other animals to stand upright and move our limbs around. So some examples of these are the kind of classic weightlifting muscles, biceps and triceps muscles that move our arms. But you can think of examples from all over the animal kingdom, like the powerful leg muscles of jumping frogs, the claw closer muscles of lobsters, and even the arms and tentacles of squid and octopi are made up of muscle as well. And of course, these are just a couple examples. So muscles are super ubiquitous across the animal kingdom, and they're used for all sorts of movements. So as you can imagine, muscle's ability to generate force and displacement has been fascinating people for a really long time. So this is a plate from the 1600s showing an experiment where a researcher has an isolated muscle that he's got held in a glass tube here. And he's actually pulling on a little string that's connected to the nerve, and he's getting that muscle to contract just by yanking on the nerve. This is a really similar preparation that I did early in graduate school. So this is another muscle from a bullfrog. We're stimulating it to contract electrically, and it's pulling on a force transducer that's up above the frame there so that we can measure just how much force it's producing. And if you can imagine looking at a muscle with completely fresh eyes as if you had never seen it before, there's probably about 1 million questions that you could ask about the tissue. So you could ask, how is that force generated at all? Where does it come from? You could ask, how is that force turned on and off? You can see this muscle is contracting and then relaxing and contracting again. Going more broadly, you could ask, why are some muscles faster or stronger than others? And kind of tied in with that, you could ask, why do some muscles have different shapes? Why do my muscles in my arms look different than the muscles in my legs? So scientists and physiologists have been pecking away at these and many other questions for a long time. And as you can imagine, that has resulted in a lot of knowledge about muscle as a tissue. But the thing that makes it an active and exciting field is there's lots of things that we don't understand about muscle. So even the best studied of these questions still have gaps where we don't really understand what's going on. So one example of something that we don't have a perfect understanding of related to muscle is where different types of muscle force come from. So muscles really generate force in two ways. Muscles generate force actively, like when you pick up a weight or stand up from a chair. Those are active contractions. Those are controlled by the nervous system, and they require energy in the form of ATP to be carried out. But muscles also generate force passively in response to stretch. So if I bend down and touch my toes, that tension I feel in my legs and in my back is from muscle, but it's not from any active contractions. It's just from kind of the natural stiffness of that tissue of muscle as a material. It's kind of analogous to how a rubber band just generates force when you stretch it out, just without any energy input. It just comes from the intrinsic stiffness of muscle. Passive force is not controlled by the nervous system, and there's no energy required for it. So we know where active force comes from. Active force comes from a complex of proteins in myosin, actin and myosin, that form these structures called myofibrils that do the actual forceful contraction. But something that I think is really surprising to a lot of people, and that definitely surprised me when I entered the field, is that where exactly this passive force comes from is still an open area of debate. So passive force could come from intracellular proteins like active force does. It could come from the extracellular matrix of collagen that holds the whole organ together. It's not really clear, and there's no kind of consensus within the field. And the problem isn't that people haven't thought of really clever ways to study this passive force. The problem is that lots of people have thought of lots of really clever ways and have come up with evidence for completely different conclusions either way. So this example kind of illustrates to me that there are really basic mechanical behaviors of muscle that we don't understand and we can't explain, even though we've been studying it for so long. So that's kind of muscle physiology as an academic field. And now I'm going to take a couple minutes to talk about hydrostatic skeletons. So when I say skeleton, probably the first thing that comes to mind are bones. So our skeletons and the skeletons of all vertebrates are made up of bone. These give us some rigidity so that we're not just a pile of mush on the floor. And they also give our muscles something to pull on so that force and displacement can be leveraged in different ways. But I think we all know that bony skeletons aren't the only type of skeleton. So another really common type of skeleton is an exoskeleton made up of chitin, like insects, arachnids, and crustaceans have. So functionally, this is the same as ours. It lends some rigidity and it gives muscles something to pull on, but obviously its design is very different. And a third type of skeleton that maybe you've never heard of before is a hydrostatic skeleton. So this is the type of skeleton that sea anemones and worms and lots of soft-bodied organisms have. And in these skeletons, there is no rigid component. There's no bone and there's no chitin. But whatever rigidity is there comes from pressurized fluid within the organism, basically pressurized water, hence the hydro in hydrostatic skeleton. So what's keeping this pink sea anemone here kind of upright is not any rigid elements within that organism. It's pressurized fluid that it's drawn in from its environment. The anatomy of these hydrostatic skeletons is pretty straightforward. All you really need is fluid in some kind of compliant container, usually cells or a body cavity of some sort. And that container has to be reinforced by tension-resisting fibers. When you put these two together, you get a composite structure that has some of the properties of both. So these skeletons can resist compression because of the fluid in there, but they can also resist tension because of the fibers. When you put them together, you also get some cool emergent properties. These skeletons can extend at really low force given the correct geometry, because the fibers that make them up can reorient and the fluid inside of them can slosh around. They're highly resistant to kinking when bent, which makes them good for really smooth and hydrodynamic body forms. And when pressurized, they can form rigid struts that are analogous to the bones or the chitin we see in other skeletons. So these are cool for sure, but what do they have to do with muscle, which is of course the subject? So I mentioned that the components of a hydrostatic skeleton are fluid-filled spaces and reinforcing fibers. And if we think about muscle kind of at the organ level, we can see that both of these structures are actually present within the tissue. So this is a diagram of muscle that shows kind of the hierarchical structure of the tissue. It's a cross-section showing a couple elements elongated out and magnified so you can see them better. Out front is one muscle fiber, which is just a single cell bounded by a cell membrane. Behind that is a muscle fascicle, which is a group of muscle fibers clumped together. And behind that is the whole muscle. So every muscle fiber is essentially a little fluid-filled bag of water, right? That's basically all cells are, just they're mostly cytoplasm. So if we wanted to make an analogy to hydrostatic skeletons, we could say the muscle fibers are our fluid-filled spaces. And what maybe you can see from this diagram is that every muscle fiber, every muscle fascicle, and even the whole muscle perimeter is reinforced by connective tissue, this white stuff in the diagram, which is collagen in the extracellular matrix of muscle. So muscle is made up of lots of muscle fibers. And if muscle is not just going to fall apart the first time it contracts, there has to be something outside of the cells that's holding it all together in a complete organ, and that stuff is collagen in the extracellular matrix. So here's what it actually looks like. These are micrographs of muscle in which all the cells have been removed chemically. All those muscle fibers are gone so that you can just see this white stuff, this extracellular matrix really clearly. So all the white is fibrous collagen, and all the black is space where muscle fibers used to be. And if you look at this bottom right micrograph, you can see the connective tissue that surrounds one muscle fiber, one single cell. you can see that it kind of wraps around the cell in a roughly helical pattern and that it connects to all the other cells around it so that it generates this kind of honeycomb network. And in these other micrographs, you can see this, the different components of this matrix just at different levels of magnification. And so if we want to continue with this hydrostatic skeleton analogy, these collagen fibers are a really good candidate for reinforcing fibers in a hydrostatic skeleton. So structurally, we've got all the components, and the question that my dissertation is asking is, do these two interact with each other and give rise to hydrostatic skeleton-like mechanical properties within muscle? And kind of the main kind of conceptual reason this is important is that when we think about muscle as a mechanical system, a force-generating little device, we typically don't think about water at all. We tend to focus on the active components of muscle, the actin and myosin, and some other spring-like elements within the tissue, but fluid just isn't really considered a mechanical component of the system. But if muscle has this internal hydrostatic skeleton, then fluid could be a really important component of the tissue, and we could be missing that. And so kind of all the research projects I've done throughout my time here have basically the approach has been to perturb the water in muscle somehow, either by increasing its volume or by pressurizing it to see if that elicits behaviors that are characteristic of hydrostatic skeletons. So the first project that I'm going to present asks, does muscle fluid volume influence that passive force that's generated in response to stretch? Does that have anything to do with the amount of fluid inside of the muscles? And this work, as Tom alluded to, was inspired by an animal that has an actual hydrostatic skeleton, which is a ribbon worm. So these ribbon worms, essentially their whole body is one hydrostatic skeleton. They have fluid on the inside and reinforcing fibers on the outside. And what Clark and Cowie showed in 1958 is that the extensibility of these worms, basically how stretchy they are, is almost completely determined by the amount of fluid inside of their hydrostatic skeletons. So worms that were really stretchy have a relatively low volume of fluid inside of their bodies. But animals with a relatively higher volume of fluid inside of their bodies are much more constrained in the lengths that they can get to. So here's a system where the amount of fluid has a big influence on the mechanics. And what we wanted to know is if this dependence of flexibility on fluid volume is a quality of muscle, as it would be if muscle has this internal hydrostatic skeleton. And so the way we did this, as Tom set up nicely for me, is we measured the passive force that muscle's generating, just the force they produce in response to stretch, before and after manipulating their fluid volume experimentally. So these experiments are done on isolated muscles that have been dissected out of an organism, a bullfrog in this case. And when we do that, we bathe them in a physiological solution that is isotonic to the muscle, meaning it has the same concentrations of salts and sugars in it, and it basically just keeps the muscle alive and happy outside of the organism. So this blue here is an isotonic solution. So one end of the muscle is held in a clamp in these experiments, stationary, and the other end is connected to a servo motor so that we can change the length of the muscle and also measure the force that it's generating at a series of lengths. So what we do is we first just measure the force that these muscles generate as you stretch them out. This is a graph of muscle length on the x-axis and muscle force on the y-axis. And what you can see is that as you get to higher and higher lengths, as you move to the right, the force that this muscle is generating gets higher and higher, just like a rubber band would generate more and more force as you stretched it out. In this bottom panel, it's still force versus length, but I've just log transformed it so you can see that information as a straight line rather than as an exponential curve, which just makes it easier to do certain analyses and statistics on. So kind of the experimental part of this is we switch out this regular isotonic solution for a much more dilute solution that is hypotonic to the muscle, meaning it's just much more dilute, much closer to water. And we let the muscle soak in this solution for 90 minutes. And what that does is it sets up an osmotic gradient, which tends to drive water from the solution towards the relatively salty muscle fibers. So osmosis is just kind of a basic physical phenomenon, and we're using that to basically inflate the muscle with excess water. And we do separate tests of muscle mass to make sure that it's actually increasing as we expect it would. And so if muscle has this internal hydrostatic skeleton, and if they're like these ribbon worms, we would expect that this increase in fluid volume would have a big influence on the force that muscles generate in response to stretch. And that's exactly what we find. So we find that if you stretch these muscles out again, after increasing their fluid volume, you get higher force generated at every length, higher passive force. So we wanted to compare this behavior of muscle to kind of a pure abstracted version of a hydrostatic skeleton. So we built one out of stuff you can just get at hardware stores. So this is the complete model here. It's made out of a flexible sleeve of plastic fibers used for organizing computer cables, and then just a flexible fluid-filled bladder there. We put those together in the complete model, and we stretch that out. And what you can see is that this model, first of all, does a really nice job of replicating that passive force versus length curve that muscles have, and also that by injecting a little bit more fluid into that bladder, by increasing the fluid volume of a little bit, we can replicate this shift in passive force. We can get more force generated at every length. So here we have an actual muscle and then an abstracted model of a hydrostatic skeleton behaving almost in the same way to an increase in fluid volume. And our big take home from this is that fluid can act as a mechanical component of muscle, right? We changed the amount of fluid in the tissue and we got completely different mechanical behavior as a result. This has been out for a while. We published it in 2017, and since then we've had lots of time to talk to people and present it at conferences. And kind of the constant question we always get when we present these results is whether or not this experiment is directly transferable to anything that happens in nature. So people want to know, do muscles actually increase in fluid volume ever, and could that affect the passive force that they're generating? So this experiment, exactly as it is, was never meant to tell us anything about anything out in nature, right? It was just supposed to tell us something about the basic biology of muscle, whether or not fluid was important. And in fact, it's completely unrealistic, because when we do this, we increase the amount of fluid in the muscle by a lot, by 40%. So imagine one of your muscles got 40% bigger in volume. That would be really noticeable. And as far as, right, as far as we can tell, that would never happen in a living organism. But we got this question enough that we dug into the literature to see if there ever are times when muscles consistently do increase in fluid volume. And we found that one time that they do that is during extended bouts of exercise. So during kind of a normal bout of exercise over the course of 30 or 40 minutes, it's actually really typical to have about a 1 to 10% increase in fluid volume of your muscles. And if you really exert yourself really hard, this can max out at about a 20% increase in fluid volume. So a lot of these studies have been done on just human subjects who are asked to cycle on a stationary bike at different levels of intensity, and then researchers just take a muscle biopsy and look at the fluid content of their muscles. And this water is coming from the vascular system. So as you work out, you get increased pressure inside of your blood as your heart beats faster and faster, and that causes some water to basically squeeze out into the muscle. Also, as your muscles do more work, you start to build up metabolic breakdown products like lactic acid, which effectively makes the muscle saltier and starts to draw water in again via osmosis. We didn't know this when we did this first experiment, but great, right? It's really interesting. So then what we decided to do is, using this same system, we decided to ask if smaller, kind of more realistic changes in volume, 1 to 20% range, can influence passive force. And while we were doing that, we wanted to see if there was a correlation between the amount of fluid you push in and the amount of tension you get out of the muscle, the amount of passive force it generates. This is work we did with Ethan Wald, who's an undergraduate who's worked in our lab for a number of years. He collected a good chunk of this data, and he helped write the first draft of the manuscript, but he's gone on spring break already, so he can't be here. So we still use a muscle in a bath, an isolated muscle. But instead of using 2 really extreme solutions for this, we use a graded series of solutions to gradually change the volume of the muscle. So we start the muscle off in just our regular happy isotonic 100% solution. We move that into a 75%, into a 50%, and then into a 25%, and we let the muscle soak in each one of these for 20 minutes. And after each soak, we measure the force that the muscle is generating, the passive force it's generating, at a predetermined length. And we also look at the mass of the muscle, which gives us a sense of how much water is moving into it. So what first I'll show you just the mass. So this is a graph with solution concentration on the x-axis and normalized muscle mass on the y-axis. And what you can see is that as you get to lower and lower solution concentrations, as you move to the right, the mass of the muscle increases, which just tells us water is moving into the muscle. And we've set it up so that we're maxing out at about 20% increase in fluid volume, which is about as much as we saw in humans at least. So next I'll show you the force that's generated by these muscles. So again, this is force generated at one kind of predetermined length that we just keep stretching the muscle to as we go into these different solutions. And what you can see is as solution concentration drops, the force that the muscle's generating at that one length gets higher and higher. And then the beauty of this experiment is you can just plot these two against each other and see that there's a really tight correlation between the amount of fluid that you push into a muscle and the amount of force you get out of it. So we fit just a simple linear model of this, and you can see it has a slope of about 2.5. So what that implies is that for this muscle, for every 1% increase in fluid volume we got a 2.5% increase in the force the muscle was producing. And so if you can take that and apply it to humans, and even though this was done in frogs, it should pretty directly translate. A pretty normal 10% increase in fluid volume during exercise should increase the force generated by your muscles by more than 20%. Passive force. And the reason that's important practically is that big changes in passive force like that could basically change the range of motion of your joints, which is partially constrained by the flexibility of your muscles. And it could also have a big influence on the energetics efficiency of movement, basically how much energy it takes to move around and stretch out those muscles. So there has been some work done in the past that's shown that really basic properties of muscle like stiffness change after people exercise, and there hasn't really been good explanations for why that is, and this could help shed some light on that potentially, these kind of fluid effects. So just to give you kind of a mechanical intuition for what's going on here, I just want to show you some videos from this model that we made, the kind of abstracted hydrostatic skeleton model. So this is the fibrous sleeve component of that model. It's just flexible plastic fibers arranged in a helix, and I think there's some of these up here. They're just for organizing computer cables. In our muscle analogy, this would represent collagen in the extracellular matrix. And what you can see is that as I stretch and shorten this model, the fibers that make it up reorient and become more or less parallel to the direction of stretch. So you can see some of them are marked in yellow, which makes that hopefully a little bit easier to see. What's a little bit harder to see is that as you change the length of this sleeve, the volume inside of it also changes pretty drastically. So if you have the model at short lengths, it has a little bit of space in there. You could fit something inside of that sleeve, but as you pull it straight, it goes basically flat, and the volume inside of there drops to 0. So this dependence of volume on length is a really basic property of these fiber wound sleeves, and it doesn't cause any problems if you have an empty sleeve full of air. But as maybe you can imagine, a problem does arise if you put something inside of that sleeve, like a bladder that has a constant volume of incompressible fluid in it. So now this is a model, same sleeve, but it's got a volume of fluid inside there. And what I want you to see is that as I stretch it out, it hits a really clear physical stop where it doesn't want to extend anymore. It goes to about 17 centimeters on that ruler there. What's happening is that as length increases, the volume of the sleeve drops, but the volume of the bladder is constant. So eventually there comes a length where these two come into conflict. And once you do that, the sleeve starts to bear down on the incompressible volume of fluid, and to stretch anymore, you either have to stretch the sleeve, stretch the actual fibers in there, which is really difficult, or compress the bladder, which is pretty much impossible because it's water. And so as you can imagine, maybe if I change the volume of fluid in there, I can change when this conflict occurs, and I can change when the model locks up and stops stretching out. So I put a little bit of fluid in, and now it'll only get to about 16 centimeters. I changed the volume of the bladder, I changed when this conflict occurs, and how much it'll stretch before it gets really hard. And just to really drive this home, if I take most of the water out, it gets really stretchy again, because now there's no conflict again, almost as if the sleeve was empty. And so this is basically what we think is going on inside the muscle. We think that as muscle is stretched out, this collagenous extracellular matrix reorients to kind of accommodate some of that stretch. But eventually, the collagen matrix begins to squeeze down on the muscle fibers and fascicles that it surrounds, and basically starts squeezing down on them and pressurizing them, so that the volume of fluid inside of those fibers ends up having a big influence on the compliance of the system overall. So I'm going to show you just one more project now, and I'm going to switch gears a little bit. So far we've focused entirely on this passive force that muscles generate in response to stretch. And now I'm going to switch over and talk about active force that muscles generate during contraction. So this is kind of a fun and strange experiment that we developed over the last couple of years, in which we squeeze a muscle with a blood pressure cuff midway through a contraction so that we can measure how squeezing and pressurization influences the active force that muscles generate as they contract. This is a bullfrog muscle again, the same muscle that we've been looking at pretty much this whole time. It's connected to a force transducer that's up above the frame that lets us measure how much force it's producing as it contracts. It's being stimulated electrically using a little electrode attached to the nerve that's associated with this muscle. And what you can see is that midway through the contraction, we're inflating this little blood pressure cuff that's wrapped around it. It's a blood pressure cuff for neonatal babies. Very cute. So I'll just let it play through a few times. There's three things that happen in pretty quick succession. The muscle contracts, the cuff inflates, and then the muscle relaxes. So here's it relaxing. So here's a contraction, an inflation, and a relaxation. So contract, inflate, and relax. Does that make sense? Cool. So at the time that the cuff inflates, the muscle is generating a pretty constant amount, kind of a steady state, what we call isometric force. It's kind of reached a plateau in how much force it's going to generate. And by inflating the cuff, when that's happening, we can see how pressurization of the muscle changes that force. And really, there's only three potential outcomes. Squeezing the muscle could increase the force that's generated, it could decrease the force, or it could have no effect, right? Pretty simple. So what we find when we do this is that squeezing does have an effect on the force that's generated, but that the magnitude and the direction of that effect varies depending on the length at which the muscle is held. So this is a graph of muscle length on the x-axis and muscle force on the y-axis, and the gray dots represent the maximum amount of force that a muscle could produce at each one of these lengths. What we found is that at short muscle lengths, on the left side of this curve, squeezing the muscle mid-contraction causes an immediate reduction in active force. So this is what that looks like on an actual force versus time trace. So this is one individual contraction here. The muscle starts off at about 0 force. It's relaxed. We stimulate it to contract. It develops force and reaches this isometric plateau. And then during that plateau, we squeeze the muscle with the blood pressure cuff right at that vertical dotted line. And we measure force immediately before and immediately after that squeeze. And that's what you see plotted on the graph.
Does the cuff remain inflated?
The cuff, yeah, remains inflated throughout. Yep. Yeah, good question. So in contrast to what we see at short lengths, at long lengths you actually see the opposite. At long lengths, squeezing the muscle midway through a contraction causes force to pop up. It's weird. It's different. So if you look at all the lengths in between these two, you can see it's actually a continuum between two extremes where the effect of the cuff goes from negative at short lengths, has no effect in the middle, and gets higher at long lengths. So this is just one representative muscle, and next I'm going to show you average data from 7 of these. And instead of showing you the pre and post force, I'll just show you the average change in force that we saw at each of these lengths. So this is average change in force that resulted during squeezing at each of these muscle lengths, and really just shows you that the effect is consistent from muscle to muscle. The asterisks denote lengths at which the force after squeezing was significantly different than the force before, and I put a 0 line in there so that you can see where the effect goes from negative to positive. And so this isn't what we expected at all when we started this project, and so the obvious question is what in the world could cause this? Why would squeezing a muscle have anything to do with the length that's being generated, or with the why would squeezing a muscle have anything to do with the length that it's at? And basically, over the course of this project, we've thought through a lot of different potential mechanisms for this, and today, for the sake of time, I'm just going to present the one that we think can actually explain this data. So as we've seen, muscles are reinforced by this really robust matrix of collagen around the muscle fibers. And we've seen that as a muscle length is increased, this matrix reorients and becomes more parallel to the direction of pull to accommodate that stretch. It's been shown that at short muscle lengths, collagen is wrapped at a pretty high angle, relatively perpendicular to the long axis of the muscle fibers, up around 75 degrees. But at much longer muscle lengths, kind of as far as you can stretch out the muscle, collagen is much more parallel to the muscle fibers, down around 25 degrees relative to the direction that the muscle fibers are going. And this is data I've pulled from a previous study that did really careful measurements of these angles as a muscle was stretched out. So the reason this is relevant is that these different angles could have a big effect on on how muscle responds to squeezing. So this is another really simple model I built that represents muscle fibers wrapped in extracellular matrix, and really it's just a fluid-filled silicone tube that's reinforced with Kevlar sewing thread in its walls. And this represents a muscle at short length. You can see the fibers are kind of packed in perpendicularly, so this is a muscle at quite a short length. And what you can see is that squeezing this model and pressurizing the fluid within it causes it to increase in length as pressure presses out on the end caps of the muscle. But if you set this model up a little bit differently so that it represents a stretched muscle with fibers down around 25 degrees relative to the long axis, hopefully you can see those. Can you see those yellow fibers in there, or is it too? Squeezing this model causes the opposite. Squeezing this model actually causes it to decrease in length. And the reason this happens is that whenever you squeeze and pressurize a fiber-reinforced cylinder like this, it always tries to reorient to a fiber angle of about 55 degrees, kind of intermediate between those two. And the reason that happens is that 55 degrees is the optimal angle for accommodating fluid pressure because it provides the greatest amount of space inside that sleeve. So remember stretching that sleeve back and forth, the angle at which the volume inside of that sleeve is maximized is at 55 degrees. So if you're above 55 degrees at 75, pressurization causes lengthening of the model, which brings it a little closer to that 55 degrees, which actually increases the space inside of there and helps dissipate some of that pressure. And if you're below 55 degrees, squeezing causes the opposite for the same reason, because moving towards 55 degrees can actually dissipate some of that pressure. And maybe you can imagine if you have a model that's already at 55 degrees, squeezing it causes pressurization but no real change in length because you're already right where you want to be. Moving wouldn't help you at all. So these are 3 models that have completely different responses to squeezing. They're all based on real muscle anatomy, just at different lengths. So I built 5 of these in addition to these. I built a 40-degree model and a 65-degree model. I connected all of these to a force transducer at constant length, squeezed them with a blood pressure cuff to see if that changed the force that they generated in a way that was similar to the muscle at different lengths. So I've got data from one muscle on the right. We've already seen this. It's just for reference. And I'll plot the data from the models on the left. One thing to note is on the x-axis on the right-hand graph, we have muscle length. And on the x-axis of the left-hand graph, we have fiber angle. And the idea is that high fiber angles correspond with short muscle lengths. Low fiber angles correspond with long muscle lengths. And that's data, again, that I pulled from some previous work. So I want to compare these two. That's why that's important. So first I'll plot the initial force that the models were generating. We set them up with a little bit of pre-stretch in them and set them at a constant length to kind of simulate them generating active force. And we looked at how squeezing them changed that initial kind of pre-tension. What we find is that at high fiber angles, short muscle lengths, squeezing causes the tensile force to drop as fluid pressure presses and tries to extend the length of the model, essentially. At low fiber angles, which correspond with long muscle lengths, we find the opposite. This one's a little more intuitive, I think. So that muscle had some pre-tension in it. We squeezed it. It tried to shrink up, tried to shorten, so you got higher tension when that happened. And if you look at all the angles between those, we can see that it moves from negative to neutral to positive, just like the muscle. So here we have simple models of muscle morphology that are able to qualitatively mimic the effects of pressurization on active force in a real muscle. And we think that this suggests that a kind of hydrostat-like interaction between muscle fibers and connective tissues could explain this effect of the pressure cuff on muscle. So I promised I would get back to how this is relevant to actual muscle physiology. So your muscles aren't often getting squeezed by a blood pressure cuff. But even though your muscles aren't getting squeezed externally by a blood pressure cuff, pressurization is a really normal part of everyday muscle physiology. So as a muscle contracts or is just passively elongated, fluid pressures develop inside of it, usually within a range of around 50 millimeters of mercury to up around 300, but it can get much higher depending on kind of what results you believe from the literature. And basically what's going on is that whenever a muscle develops force of any sort, because of the complexity of the shape, some of that force is directed inwards so that it ends up squeezing the muscle effectively and pressurizing the fluid within. So just like kind of most models of muscle don't really consider fluid volume and how that affects the mechanics of muscle, they also don't think about the pressure that influences muscle and how this could influence the force that your muscles are actively generating out in the world. But what our results are suggesting is that the state of pressurization and the distribution of fluid within muscle can have a really significant influence on contractile force. And since your muscles are always pressurizing whenever they contract and whenever they're extended, these effects that we see amplified by the blood pressure cuff could potentially always be at play, just at different levels, kind of whenever your muscles are contracting or being elongated in real life. So to kind of summarize and wrap up, I'm going to return to this initial question of, does muscle have an intrinsic hydrostatic skeleton that influences its mechanical behavior? So showed kind of implicitly throughout the talk that muscle has the necessary structures for a hydrostatic skeleton. It's got fluid-filled spaces that are reinforced by fibers. I showed you that the passive force that muscles generate in response to stretch is influenced by the fluid volume in a way that's analogous to an animal that actually has a hydrostatic skeleton and to an abstracted model of a hydrostatic skeleton. And then finally, I showed that that active force is influenced by internal pressure in a way that really only makes sense in the context of a hydrostatic skeleton. So does muscle have an internal skeleton? We think all this evidence indicates that yes, muscle has a soft skeleton made up of fibrous collagen and fluid-filled muscle fibers. And we think that this concept of muscle as a hydrostatic skeleton that's also actuated by these active components has a lot of potential to help us understand some fundamental behaviors of muscle that have kind of eluded physiologists for a long time. Okay, science is over. Thanks for hanging in there. I have a lot of people I want to thank, and before I do anything else, I just want to say that even though, so my name's on this obviously, but none of this gets done in a vacuum, and I just want to thank everybody who helped on this project, either like directly contributing to it or just talking about it, because all conversations help when you're trying to figure something out. And I especially want to thank kind of the people in the Roberts Lab who've been affiliated with the Roberts Lab kind of in my time and before I was here, because without kind of the work that these people were doing before, I just wouldn't have done this project. It wouldn't have turned out this way. So thank you to everybody. I also want to acknowledge that, I wouldn't have been in a position to enter grad school or write a dissertation without the support and encouragement of my family, who have always encouraged me to pursue education. So thank you very much for that. And also thank you for coming. It's super fun. It means a lot. I also want to thank Casey for just support on a daily basis, especially in these last couple months of stressful finishing up. Thank you. I also want to thank our cat Arden for being an awesome distraction from work and for just being a source of fun. I want to thank Chris Stover and Pete Feltman, who were kind of some of my best, earliest friends when I moved to Providence, and they really made me feel like I had a home, away from Michigan, so that really meant a lot to me. I want to thank my committee, so Tom, Beth, Rich, and Steve, for giving me really thoughtful and critical feedback throughout this whole thing, and for just being really good science mentors. And obviously I want to thank Tom for just being a super caring and thoughtful mentor. So thank you so much. I want to thank my earliest mentors at Michigan State, so Susan Hill and Richard Hill kind of helped me find a foothold in science and also kind of helped me realize that grad school was the thing for me, so I wouldn't have been here kind of without their help. And then there's lots of people at Brown I want to thank, so the whole EB department in general. Just thank you for kind of facilitating a happy and collaborative environment. I want to thank Sharon for having me as a TA in biomechanics early on, which was super formative and important. The whole EEB staff for supporting me and other grad students kind of throughout our careers, and Leanne has been especially helpful as I've been navigating finishing up. Erica keeps all the labs running. I don't see her, but thank you. And also Kim has been kind of my go-to person throughout this whole thing, so thank you to both of them. I want to thank the animal care staff and veterinarians for allowing the animal work to happen. Other people at Brown, are Jeff Williams and Justine Allen, kind of helped me with the technical aspects of the microscopy that I wouldn't have figured out without them. And also Mike and Dale made the anatomy lab just a super fun place to be. So thank you for that. I really, really enjoyed my time in the anatomy lab. This also wouldn't have happened without funding. So these are the different organizations and groups that funded this work. And without these and without the people that contributed money to them, it just would have been ideas. So thank you to these organizations. And then last but not least, obviously, so I just want to thank all the EEB grad students and postdocs that have made this just such a fun time. I've really enjoyed my time in Providence, and that's all because of the people that I've gotten to spend my time with. So thank you to all of you. Even if I didn't have a picture to put up here of you, thank you for being part of this super fun community. And that's it, so I'll take any questions.
Thanks, David. We have time for questions. Typically, the grad committee members will ask their questions in the closed door session afterwards, but we're open for questions from the floor. Just a quick announcement. There's a celebration tonight at my house at 6 p.m., 24 Short Road. I'll write the address on the board. Hopefully you all got the email, but I hope many of you can make it.
Stephen?
So thank you for a wonderful presentation. It's really fantastic, and I learned a lot. I have maybe two related questions. The first one is about all of the microscopy of collagen that you showed us was sort of, I don't know what the right word is, end on. Mm-hmm. But then the Kevlar thread experiment showed that the effect of compression depended on the sort of angle of the fibers, which you couldn't see in the collagen.
Right.
So I guess my question is, do different muscles in different parts of the body have different orientations of the collagen fibers in 3 dimensions that affect what happens to them when they get squeezed?
Yeah, I don't know because there's not data on that, but almost certainly, yes. That's I came in really interested in how muscles become specialized for different tasks, either force production or dissipation of energy, and that's kind of, we didn't even know where passive force came from, but as I've been working on this, it seems like, yeah, probably these different orientations of collagen could have a big effect on that. And even in with age and in neuromuscular disorders, your muscles get stiffer, and it's not really clear why that is. And it's been shown that it's not the amount of collagen that's changing, and so I think that it's the arrangement of collagen is getting messed up. Yeah.
That actually leads to my follow-up for all of the aging athletes in the room. Everyone's trying to sell compression socks and sleeves and everything.
Yeah.
So should we get those?
The pressures that a compression sleeve generate are not as high as what we did here. So
I'll save my money.
Yeah. People think they could help with like blood return or with proprioception of some sort, but probably not for this kind of stuff. Yeah. Thanks. Dale?
So maybe then following from that, is it possible that the collagen angulation can remodel in response to, I don't know.
Probably. I mean.
Or is it set genetically?
I don't know. That's a great question. I mean, nobody knows. What I would guess is that I think that these structures are laid down in response to mechanical stresses.
Okay, yeah.
So, one example that comes to mind is a contracture that is a muscle disease where you have a limb often that's stuck up like this, and that's because your muscles are way tenser than they should be. And I would guess that's kind of a feedback thing where you have some tension on a muscle and it's generating too much collagen, something like that. But
those angles have been measured?
In contracture people?
No, in muscle.
Yeah, the angles have been measured in muscle, but only in a few studies.
Okay. So you don't have enough to know to say definitively if they vary within muscle between muscles.
But they do turn over. Like there's a lot of degradation of collagen going on all the time and laying down of new collagen, so it has the potential to remodel. It's not set from birth. Yeah.
Oh, that's cool.
Giordano, hey.
Hey, that's awesome, dude.
Thanks.
So I was just wondering, if you started thinking, because you're answering some really fundamental questions about the effects of pressurization and the role it has in muscle generating force. And so I was wondering if you started to think about how partial activation of a whole muscle belly might affect force generation. So if you have like a sausage of muscle and only parts of the inside are becoming active, so not the muscle isn't fully active, how that might affect the generation of force, and it's how that's impacted by things like the sleeve of tissue around it.
Yeah, super interesting. Yeah, I mean, basically, if you had the core of the muscle active and the rest of the muscle passive, it would be almost like you had a little sock around that portion of the muscle, right? And so you might get higher pressure in there. What I've tended to think about is different parts of the muscle being differentially activated and squeezing different parts. So kind of the same idea, just in reverse, could have a big effect in ways that nobody's ever really studied. Bianca?
Why frogs?
Oh, that's a great question. So frogs are just kind of a classic, traditional model that's been used in muscle physiology for a long time. So we just know a lot about how their muscles work, kind of how they should behave. They have some muscles that are really nice and simple in their architecture. So we picked the semimembranosus muscle. It's not this muscle. This is actually a different one. But the one that all my experimental work was done on is a muscle with it's basically just, it's like a strap. It's just muscle fibers going straight from one place to another. And we chose that because it's a little bit simpler than trying to understand how something like a more complex pennate muscle or something with multiple tendons would respond. So frogs, because they're easy to house and maintain, and they have big muscles for a relatively small animal, and because they have muscles that are kind of shaped the right way. Stephen, hey.
Hi. I was wondering if your model was relevant to organisms in nature that, say, lose water due to water limitation or desiccation or like cholera-like diseases, and how much and the related question is, do you know much about how much water can be lost from a muscle during those kinds of stressors?
Yeah, I would love to know. I've kind of looked in the literature and haven't found a ton of information. There's a little bit of work on humans and dehydration and how your muscles change during dehydration. I don't.
Right now?
Yeah. So as you sweat, you're losing water, and yeah. I can't think of a figure off the top of my head for how much volume change you get. Whenever we've thought about volume changes in our experiments, we've thought about increasing it, partially because if you try to go the opposite way and you soak the muscle in a hypertonic solution that's much more salty, it starts to kind of twitch erratically. So it's affecting the biochemistry of the contractile proteins somehow, and that kind of throws off your ability to learn anything about, be confident in that you're measuring passive force. So experimentally, we haven't really been able to touch that. But if there were, desiccating worms or something, that would, I imagine that would totally throw off of their muscle physiology, and I would love to find an example of that. Yeah, that'd be cool. David Burma.
I wish I could make this question a little bit more concrete, and maybe I will later, but do you have a sense for how like in-series elastic elements like stretchy tendons might interact with the muscle belly itself to modulate the length of the whole MTU when muscles are being pressurized or changing fluid volumes and things like that?
Well, so we know that kind of you can have muscle shape changes that are not reflected in the length of the whole muscle tendon, because basically a muscle can stretch against a tendon, but how these kind of fluid interactions would come into play, I'm not sure. Aaron? Hey.
Oh, great talk. That was so cool. Maybe this question doesn't make any sense, so that caveat. You're changing if you're talking about changes of the volume, that could change the orientation of the fibers, and that's as a result of changing the volume. But what about active contraction and changing the shape of the muscle? I recognize that's isovolumetric, which is departs from like the first model where it's not isovolumetric. But when you get reorientations of the fibers with active contraction, and how might that then relate to force reduction over the like tension kind of curve?
Yeah, so that gets at a really fundamental idea that we haven't quite been able to figure out how to touch experimentally, but it's just conceptually really interesting, which is, typically when we think about force generation, it's generated by these myofibrils and it gets transported linearly out to the tendon, just kind of along the chain. But here, you could have a potentially different route where that myofibril force goes into pressurizing the fluid, which then gets pushed out on the extracellular matrix, and then it gets transferred through the extracellular matrix through the tendon. So if you're familiar with a McKibben actuator, it's basically that, but in a muscle. But how to kind of attack that experimentally, we're not sure how to do that yet. But it would be super cool, because one extreme would be all force goes through that route, and the other extreme would be no force goes through that route, and it's probably somewhere in the middle.
Okay, well, let's thank David and